Hello everyone, I am Pallavi and on this channel I will be guiding you to the journey of class 9th and 10th science in the simplest way possible. So let's begin with the very first chapter, that is heredity. From the color of your eyes to the way you smile, heredity connects generation. So in this chapter we have to cover the following topics. We'll begin with the key concepts of this chapter and then accumulation of variations, acquired and inherited traits, and very important, Mendel's experiment and the laws of inheritance. Then we'll discuss how blood groups are inherited, and the last part will be the gender determination. So let's begin with the key concepts of this chapter. First, we'll discuss what is heredity. So, heredity is the transmission of characters from the parents to the offsprings. Now, character word could be replaced and traits could be used. So traits and characters means the same thing. Now, how the hereditary information passes from the parents to the offspring? This is because of the gametes. And we all are aware about what are gametes. So we can say that gametes are the link between one generation to the next. The second part of the key concept is variations. So, now what are variations? Variation is the difference between the characteristics among the individual of a species. And we all can see variations among ourselves. We have different heights, we have different eye color, and one example which is in our NCRT also, free earlobe or attached earlobe. So many a people have free earlobe and a few people also have attached earlobe. So we can write that what are variations? Variations are the difference in the characteristics, or we can use the word traits, among the individual of species. The individual of a species. And always remember, variation is the necessity for organic evolution. So it is the necessity for organic evolution. So the third key concept is to learn about what are chromosomes and what are genes. You are aware about the chromosomes. You have discussed this term in class 9th also. So the chromosomes are present in the nucleus of the cell. So if I draw a enlarged chromosome here, what is a chromosome? Chromosome is the condensed chromatin material, the condensed DNA threads. So if I stretch a DNA segment here, so particular DNA segment will be called as gene. So this is my chromosome. This complete part is my DNA, and a segment of DNA is called as gene. So gene are present on the chromosomes. A chromosome have multiple genes. For example, this is a gene for my height. So this could be the gene for my eye color as well. So I can say that gene is the actual unit of heredity, which will transfer the characteristics from the parent to the offspring. Now we should discuss more about gene. So gene always occurs in pair. For example, if I am having a gene here for the height, so I will also have the same gene here located at that side only. Now gene are of two types. Let us discuss the two types of genes: dominant gene and recessive gene. So what are genes? You are aware of this thing. Genes which will decide your appearance, which will be able to express itself, which will be able to express itself, is my dominant gene. Dominant gene is always written with a capital letter. On the other hand, what is recessive gene? Recessive gene will only be able to express itself when an identical gene is present with it. So it will only be able to express itself with identical gene. It will be represented by a small letter. Let us take an example of the height of gene. You will be able to understand it much better. So, with capital T, I will represent my dominant gene. With a small letter, I will represent my recessive gene. So, this is my dominant gene, and this will be my recessive gene. Who will be able to express itself? The dominant gene. Recessive gene will be only able to express itself when it is present in pairs. One more thing, these two contrasting pair of the characters of gene are called as alleles. So we are saying that every individual are having two copies of gene which are called as alleles. Now coming to the traits, we have two types of traits, homozygous traits and the heterozygous traits. Let us discuss. We know that the traits and characters means the same thing. For example, my height will be a character, a trait. So I can have two conditions of traits which is homozygous and heterozygous. When I will have two same kind of alleles, both of the alleles can be dominant, both of them can be recessive as well, but they should be same. They will be categorized as homozygous. For example, if I'm writing capital T, both of the alleles are same and they are representing a dominant gene. So the organism will be homozygous dominant, or we can say homozygous tall condition is there. If I'm writing small t, again the condition is homozygous, and now in this case it is showing recessive gene. So it is homozygous recessive, or we can say homozygous dwarf individual is formed. Now what is heterozygous? When two different type of alleles are there, two different alleles are there. Now we'll write an example for the same, that is capital T, small t. This is the only condition which we can see in the heterozygous condition. So the organism will be, who will be able to express itself? Which gene will be able to express itself? The capital one, which is the dominant one, which is tall. So the condition will be heterozygous tall or heterozygous dominant. There is no as such condition of heterozygous recessive. You all are aware of this thing because we have only discussed the recessive condition is only when we have similar types of allele, and both the alleles, both the genes should be what? Both the genes should be recessive. So the last two key terms are phenotype and genotype. What is genotype? Genotype is the description of genes present in an organism. And what is phenotype? The characteristic which is visible in an organism. For example, in the pea plant, the plant height can be tall or the plant can be dwarf. So this too will be the phenotype of the plant. And what will be the genotype? For the tall, we can have the set of capital T, dominant, homozygous tall. Or small t, which is recessive, homozygous recessive or homozygous dwarf, and capital T small t. The phenotype will be tall, but the genotype is heterozygous dominant condition. So now you know the difference between a genotype and a phenotype. Let's begin the chapter and let's come to the second part of this chapter. Coming to the accumulation of variation. Now we know what is variation. Reproduction causes variations also, and variations are very significant for the survival in the changing environment. We need variation, but variation is of no use if it is not passing to the next generation. अगर वो पास ही नहीं हो रही है coming generations के साथ साथ, तो वेरिएशन हमारे लिए यूजफुल नहीं है. So variations produced in an organism during successive generations get accumulated in the organism. Significance of variation shows up only if it continues to be inherited by the offsprings in several generations. So we know what is variation, we know the significance of variation, and we know how variations are accumulated to the generation. Now we'll learn about what are acquired traits and what are inherited traits. See, if I'm learning something in my lifetime, if I learn a new language, suppose I learn Punjabi, will it be the case कि जो मेरे बच्चे होंगे उनको पैदा पैदा होते ही they will be able to speak Punjabi? No. अगर मैंने ताइक्वांडो सीख लिया तो क्या ऐसा होगा कि जो मेरी कमिंग जनरेशन होगी, मेरे ऑफस्प्रिंग्स होंगे, वो भी ताइक्वांडो सीख के आ रही आएंगे? नहीं ना, बिकॉज आई हैव लर्न दिस स्किल्स इन माय लाइफ टाइम. सो दीज आर माय acquired traits. So what are acquired traits? The traits which you acquire during your lifetime. अब क्या ये ट्रांसमिट होंगे मेरे कमिंग जनरेशन को? नहीं. तो व्हाट आई विल राइट दैट दे आर नॉट inherited nor they are transmitted. And what will be the example of acquired trait? Any skill which you learn in your lifetime. Suppose you learn a new language. Suppose I get a tattoo on my arm, tattoo on my wrist. So that will be acquired trait. I got ear piercing. That is also acquired trait. Coming to what are inherited traits, the traits which are transmitted to the next generation, which will I get from my parents and I will further transmit them to the coming offspring. So the inherited traits are the traits which are inherited from our parents. They will be transmitted to the next generation as well. So yes, they are inherited as well as will be transmitted to the next generation. And what will be the example of inherited trait? Blood group is an inherited trait, yes or no? Any other example you want to give? Color of our eyes is an inherited trait, and so we have many examples of the same. Coming to the next, हमने एक एक्सपेरिमेंट किया. हमने क्या किया? वी टुक माइस एंड हमने उनकी टेल्स कट कर दी, तो हमारे पास टेललेस माइस आ गए. अब हमने क्या किया? उनको ब्रीड करवाया और इन द प्रोसेस ऑफ रिप्रोडक्शन उन्होंने जो ऑफस्प्रिंग्स दिए, जो रैट्स दिए, वो टेललेस होंगे और उनके पास टेल होगी. दे हैड टेल। तो ऑफ कोर्स अगर मैं लाइफ टाइम में मेरा अगर हाथ एम्पुलेट हो जाता है, इफ माई लेग इज एम्पुलेटेड, तो देयर इज नो चांस कि माई ऑफस्प्रिंग विल ऑल्सो हैव अ एम्पुलेटेड आर्म और लेग. So it is my inherited trait because so the tails of the mice was also a inherited trait. हम लाइफ टाइम में कोई ऑर्गेनिज्म की बॉडी में कुछ भी चेंज करते हैं, दैट इज अ एक्वायर्ड ट्रेट एंड दैट विल नेवर गेट ट्रांसमिटेड टू द नेक्स्ट जनरेशन. Moving to the very important part of this chapter and the main part of this chapter, Mendel's experiment and the laws of inheritance. In the starting of the chapter, I have told you that who was the father of genetics? It was Gregor Mendel. तो ग्रिगोर मेंडल was the first scientist जिसने systematic तरीके से inheritance को study किया. मेंडल के टाइम पर क्रोमोसोम्स और जीन्स किसी को नहीं पता थे. क्रोमोसोम्स एंड जीन्स वर नॉट, even the words were not discovered at that time. तो उसने जीन्स की बजाय उसने वर्ड यूज करा था फैक्टर्स. सो फैक्टर्स कुछ नहीं है, फैक्टर्स क्या हमारे जीन्स ही है. अब मेंडल ने किस पर एक्सपेरिमेंट किया था? उसने पी प्लांट लिया. पी प्लांट का साइंटिफिक नेम क्या है? पाइसम सटाइवम. ये तो पाइसम सटाइवम, उसके 7 contrasting characteristics he studied on them. So what were the 7 characteristics? He experimented on the height of the plant height, the color of the flower color, the color of the seed, so seed color, seed shape, then the pod color, pod shape, and the last was the location of the flower, position of flower. अब ये जो genes है, we have already discussed that in the genes we have a dominant gene and we have a recessive gene. So let's see कि plant height में dominant gene क्या होगी. हम इतनी देर से examples पढ़ रहे हैं कि dominant gene plant height में क्या होती है? It is tall. And what is the recessive trait? It is dwarf. फिर आया flower color. फ्लावर कलर जो पी में होता है विच इज डोमिनेंट इज वॉलेट कलर एंड रिसेसिव इज वाइट. कमिंग टू द सीड कलर. अब देखो सीड कलर जो डोमिनेंट है पी में दैट इज येलो और रिसेसिव कौन सा है? ग्रीन. सीड शेप डोमिनेंट है राउंड सीड और रिसेसिव रिंकल्ड. पॉड कलर जो पी का छिलका है उसको बोलते हैं पॉड. सो द पॉड कलर विच इज डोमिनेंट इज ग्रीन और इसमें उल्टा है, रिसेसिव क्या है? येलो कलर. Then we have the position of flower. The position of flower which is dominant is axial and the recessive is terminal. तो अगर मेरे पास ये मेरा प्लांट है, मेरा अगर फ्लावर यहां पर आ गया है तो दिस इज माई व्हाट? माई टर्मिनल flower. And if my flowers are on the axial side, that will be the dominant axial part. Now you know the different traits which the Mendel took for the experiment. We should also learn कि उसने pea plant को ही क्यों choose करा. Why he chose pea plant? Why pea plant? So we have few advantages of taking pea plant. And this question is also asked in your boards as well. So let us see कि pea plant क्यों लिया उसने. He chose pea plant for the following reasons. सबसे पहला that the pea plant have a very short lifespan. So we can study multiple generations in a very short lifespan. दूसरा that it was a annual plant. तो flowering एक साल में multiple times होगी. And then it was a bisexual flower. So the male and female part of the flower was present on the same plant. अब bisexual flower है तो we had the option, we had the choice for cross pollination as well as self pollination. Then it had seven contrasting traits. We have already discussed about them. And the last one was that large number of offsprings were produced after fertilization. So these were the advantages of selecting pea plant. Now, what does Mendel did? Mendel ने बहुत सारे hybridization experiments perform करे. We will discuss कि उसने कैसे उन experiments को perform किया. तो इस चीज में we have to study about monohybrid cross and dihybrid cross. So let us start with monohybrid cross. देखो monohybrid में mono word. What does mono word say? Mono word means single trait. So in the monohybrid cross, he studied about a single contrasting trait and how the trait is being inherited. तो monohybrid cross को बनाना सीखते हैं. तो first we are going to discuss about monohybrid cross. Mendel selected the trait of plant height. Plant height में we know that the dominant trait is tall plant and the recessive trait is dwarf plant. So he chose two parents. तो monohybrid cross बनाने के लिए first step is to write the parents. He chose pure breed of tall plant and a pure breed of dwarf plant. Pure breed tall and pure breed dwarf. And how do we write? We know how to write the genes of the same. Capital T, and small t for the recessive trait. And we will put a cross between this. So that we are showing that they are crossing over, they are breeding. After the parents, we have to write the gametes. अब यहाँ पे genes जो है, they will segregate. एक गेमीट में एक जीन, दूसरे गेमीट में दूसरी जीन. Similar with this also. One gene will carry one gamete will carry one gene. Now we'll form the F1 generation, the first generation. Filial generation भी we can say this. अब क्या होगा? There are probabilities that this gamete will combine with this one or this gamete will combine to this one. Similarly, this gamete again can fuse with this one और ये भी क्या करेगी? इसके साथ ग्रुप फॉर्म कर देगी. तो जितने भी F1 जनरेशन आई है हमारे पास, सबका फिनोटाइप तो क्या होगा? They all will be tall. बट उनका जीनोटाइप क्या है? एक कैपिटल टी एक स्मॉल टी. मीन्स टॉल है बट हेट्रोजाइगस कंडीशन में. इसके बाद मेंडल ने क्या करा? जो F1 जनरेशन थी ना, उसकी सेल्फिंग करवाई. तो मतलब F1 जनरेशन से दो ऑफस्प्रिंग्स को लेकर उनके बीच में क्या करवाई? सेल्फिंग. तो अब हम इसके बाद लिखेंगे दैट सेल्फिंग बिटवीन F1 generation. अब कैसे बनाते हैं हम एक पुनेट स्क्वायर? मतलब F1 जनरेशन में जो सेल्फिंग हुई है हम उसको भी ऐसे ही रिप्रेजेंट करेंगे बट एक डिफरेंट तरीके से. उसको बोलते हैं पुनेट स्क्वायर. नाउ विल लर्न हाउ टू ड्रॉ पुनेट स्क्वायर. सो सेल्फिंग बिटवीन F1 जनरेशन करवानी है अब हमने. तो अगेन द फॉर्मेट इज सेम. फर्स्ट विल राइट द पेरेंट्स. अब F1 जनरेशन में तो पेरेंट्स सेम थे तो बोथ साइड विल राइट कैपिटल टी स्मॉल टी, कैपिटल टी स्मॉल टी. और बीच में क्या ड्रॉ करेंगे हम? एक क्रॉस. देन विल राइट दी गैमीट्स. तो गैमीट्स कैसे बनेंगे? यू ऑल आर अवेयर ऑफ दिस थिंग. वन जीन वन गैमीट. सो कैपिटल टी स्मॉल टी. टू गैमीट्स कैपिटल टी स्मॉल टी. अब हम पन्नेट स्क्वायर में क्या करते हैं? टू get the F2 generation, which is filial 2 generation, will draw a square. On one side we will write the male gametes and the other side we will write the female gametes. So there is one and two gametes this side, one and two gametes this side. अब क्या करेंगे, हम इनकी क्रॉसिंग करवाएंगे, कैपिटल टी, दिस टी विद द स्मॉल टी, कैपिटल टी, स्मॉल टी एंड स्मॉल टी. So this progeny will be tall and the condition is homozygous tall. This will be tall but the condition will be heterozygous tall, similar with this one, and this one will be dwarf, homozygous recessive dwarf. So let us now write the phenotypic ratio and the genotypic ratio. You are already aware of this thing, what is a phenotype and what is a genotype. So let us write phenotype was the visible trait. So here we have two traits, one is tall, one is dwarf. So how many progeny are of tall height? 3. And how many are of a dwarf height? 1. So the phenotypic ratio is 3 raised to 1. 3 are tall and 1 is dwarf. We can also represent this in the form of percentage. So it will be 75% ratio 25%. Easy. Writing the genotypic ratio. Genotypic was the genetic composition. So here one is homozygous dominant. So one homozygous dominant, two are heterozygous dominant. So two heterozygous dominant, and one is homozygous recessive. So we can also write it like this, that one is capital T, two progeny, two offsprings of a capital T small t, and one is of small t. This is homozygous tall. This will be heterozygous tall. And the last one will be homozygous recessive. So these were the observations of monohybrid experiment done by Mendel. In the second part, we will continue with dihybrid cross and the rest of the concepts of the chapters. Also, you can download my handwritten notes. I have attached them in the description box. Thank you so much.